Can somebody confirm you can hear me?
I can hear you.
Thank you. Let's see. I think we're on chapter 13. All right, sharing my screen. Should be able to see my screen now. I think that might be right. All right, today is June 6th, and we should be on chapter 13. We should be able to do, wait a minute. We should finish chapter 13 today. There is no lab today. I will be there for the first 15 minutes to answer questions. Is there any question anyone has? And my throat is a little sore again, not sore, but hoarse again today. So I'll try and speak softly and get a little closer to the microphone and see if that helps. If I'm getting too quiet, please let me know. Like I said, my voice is hoarse, so I'm not going to be speaking up today. Any question about anything? If not, let's go to chapter 13. Oh, I think I'm shared my screen. So let me state that I did do a fair amount of grading. I think I have all the labs graded for 2 times except for lab 9. So I believe I finished maybe lab 6, 7, 8. So they're graded 2 times. And over the weekend, I believe I graded all of the extra credit study groups. Any question about any of that or any of the grading or anything else, I guess? If not, let's go to viruses, chapter 13. We had talked about the multiplication of animal viruses and mentioned that they largely follow the same pattern of replication of bacteria viruses, meaning they have to infect a host cell, attach to a host cell, infect the host cell, replicate, and then be released. And how they replicate usually is they take over the cell's machinery and use it to replicate the virus. Now, with animal viruses, not all animal viruses lyse the cells, and we'll talk more about the different life cycles of animal viruses. There are some animal viruses that do lyse cells. We call them lytic viruses, but there are other life cycles with animal viruses. The main difference between animal viruses and bacteria viruses is animal viruses can replicate in the nucleus, and there is no nucleus for a bacteria virus. So let's talk about the multiplication of animal viruses. They can lyse cells, in which case they will rupture the cell. This is usually done by a non-enveloped animal virus. And this is the lytic life cycle. It follows the same life cycle, the lytic life cycle of viruses for bacteriophages. There is another life cycle, a major life cycle for animal virus, and these we call the budding life cycle, where the virus buds out of the cell. This happens in an envelope virus. It usually does not kill the cell. If the cell dies, the cell usually is killed by the host immune system, recognizing that that cell is infected with a virus, and then the host immune system kills the cell. Any question about any of that? All right. How a budding virus works is the virus replicates. Let me see if I can blow this slide up. The virus will replicate, and this viral replication will be at low numbers so that large numbers of viruses are not made by a budding virus usually. And when the virus makes a copy of itself, it will come up to a membrane. This membrane can be the plasma membrane as shown here, but it could also be the nuclear membrane or the membrane of the endoplasmic reticulum. It just depends on the species of virus, which membrane it'll bud out of. And then the virus puts pressure on the membrane, budding it out, come on, and then puts more pressure on the membrane. And then this breaks right here, and this is the cell membrane, let's say, will reform, and then that'll make almost like a vesicle around the virus, but that's not a vesicle. That is actually the envelope of an enveloped virus. And that is how the envelope viruses obtain their envelope. And that is they actually bud out of a membrane of a cell. In this case, we're showing it as a cell membrane, but they could be the nuclear membrane or the membrane of the endoplasmic reticulum, depending on the species of virus. Any question about any of that? So all of the envelope viruses follow the budding life cycle, and those are mostly herpes viruses and HIV. I'm sure there are other budding viruses, but I just don't know off the top of my head. I should know because when we think about what are envelope viruses, there's a whole bunch of human viruses which are envelope. What is that disease called? Rabies is an envelope virus. And that's how the virus obtained its envelope. You'll note that there are viral proteins or glycoproteins put into the membrane by the virus. And the virus will come up to the membrane where the viral proteins or glycoproteins are. And then when it rips off a portion of that membrane, that envelope will have the viral proteins or glycoproteins in it. And that could be like the spikes of an envelope virus. Any question about any of that? Here's an actual electron microscopic image. This is a cell right here. And these are viruses budding out of the cell. These are other cells here. They're hard to make out where the cell is, but that's clearly one cell right there. And that's clearly one cell right there, but the bud's very obvious right there. All right, viruses have different genomes. You already know that from the bacteriophages, but their genomes can be double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. And remember, if we're talking about single-stranded RNA virus, you need to state whether that's the positive single-strand RNA or the negative single-strand RNA. Therefore, each virus type needs to have a slightly different mechanism for multiplication and biosynthesis. Replicating the double-stranded DNA is very easy. You just separate the two strands of viral DNA and then use one strand as a template to make the other strand. And actually, it's the same way double-stranded RNA is replicated. It's just that RNA is the template instead of the DNA. Any question about any of that? All right. I don't know why I'm a little bit chilly. I've actually got the window open. It's supposed to be 80 degrees outside, but for some reason I'm a little chilly. Oops, let me blow that up a little bit. This is showing you the DNA family of viruses that cause human diseases. And as you can see, there are 7 families. 7 families of DNA animal viruses that cause human diseases. Now realize there are more DNA animal viruses, but some of the families don't cause human diseases. We'll talk about a few of the families. Poxviridae includes the smallpox virus. We'll talk a little bit about that. Herpes viridae, we've already talked a little bit about that. That's the herpes family of viruses. We won't talk about the papilloma viridae. Polyoma, sorry, I can't read that. Polyoma viridae, adenoviridae. I'll talk a little bit about that one. The reason is that this family of viruses actually causes the human colds. However, there are 3 families of viruses that cause human colds. So not one species, but actually 3 different families. One of them is the DNA animal virus, and it's actually the 3rd most common, or I could say the least most common cause of human colds. Hepatidinoviride is actually the virus that hepatitis B is in. We really won't talk much about that. And paroviridae, we won't talk about that either. If you know the 5th disease, it's caused by this family. Any question about that? You don't need to memorize this slide. All I'm doing is showing you the different families of DNA animal viruses that cause human disease over here. And then it's showing you the type of DNA, double or single, and whether the virus is enveloped or non-enveloped. Smallpox is enveloped virus, and I knew the herpes viruses were enveloped. I did not remember that the hepatitis B virus was enveloped. You don't really need to know the capsid symmetry. I'm not going to talk about that, nor do you need to know the size. Like I said on this slide, you really don't need to know much of this slide, okay? We will talk a little bit about smallpox, and it belongs to the Poxviridae family. And then we'll talk a little bit about, we've already talked about the herpes viruses belonging to the herpes viridae family. So here's an adenovirus, and this is a naked virus having capsomeres there. And this is a herpes virus. Let's see if I can push that up. And there's the capsid, and it's surrounded by an envelope. For DNA containing animal viruses, and this is actually, well, we're only talking about animal viruses, but for DNA animal viruses, if they contain DNA, then they generally are copied in the nucleus. And they usually use viral enzymes. But the capsid and the proteins of the viruses are synthesized in the cytoplasm using the host cell enzymes and the host cell machinery. So a rule of thumb is for a DNA animal virus, it replicates in the nucleus. And I don't know of any exceptions to that rule of thumb. Come on. Once viral DNA enters a cell, one of 2 things happens. First, the viral DNA will enter the host nucleus. The viral DNA can be expressed, producing new virions, and then that cell will um, make the virions be released. If they're a budding virus, the cell will release the viruses by budding. If it's a lytic virus, the virus will lyse the cell and that will release the new virions. But the second thing that can happen with the DNA animal virus is the DNA can enter the nucleus, and then the viral DNA can integrate into the host cell's chromosome. This we call a provirus. You can't call it a prophage because a phage is a bacteriophage, and so we call it a provirus. When the DNA of an animal virus integrates into the host cell chromosome, it will remain a permanent fixture of that host cell's DNA, meaning that rovirus will never leave that host chromosome for as long as that cell remains alive. And then the provirus will only be replicated when the host replicates its DNA to make two cells. Any question about any of that? For a DNA animal virus, the double-stranded DNA has to be replicated, making new double-stranded DNA. And that's true for single-stranded DNA or RNA as well, but we're only going to show it for double-stranded DNA because I'm just trying to show what happens to produce a virion. And then the DNA will be transcribed into viral messenger RNA. That will leave the nucleus, because remember, this is a DNA animal virus replicating in the nucleus, go into the cytoplasm, be translated into viral proteins, including the capsid proteins. The capsid proteins will then self-assemble, get together with the newly replicated DNA viral genome to make the new progeny virions. Any question about any of this? All right, let's go into the multiplication of a DNA virus in more detail. Let me blow that slide up. So we have a DNA animal virus, and this is a naked virus, but it could be the same for an envelope virus. It has to attach to the cell membrane of the host cell. Once again, there will be specific ligands or adhesion molecules on the virus that will bind to specific receptor molecules on the host cell. And that will determine what cell that this virus can bind to. And viruses, animal viruses, are just as specific as bacteriophages, meaning they generally only infect a few cells of one or a few species. We'll talk about HIV later. HIV, I just am mentioning this one because I know it, can only infect a few cells of two species. Obviously, it can infect the human species, and it only infects a few cells of humans, like it can infect T helper cells, it can infect macrophages, it can infect brain cells, it can infect a few other cell types, but it does not infect most cell types. It does not infect a bone cell, it does not infect a muscle cell, doesn't infect skin cell, okay? And it only infects two species. One species of chimpanzee, I don't remember, it might be the chimpanzee, is it bonsai or bonzi, something like that. Not chimpanzee troglodytes, if you know the chimp species. But, and then the human sapien, homos, humans. The point is, it's very specific. And that specificity depends on the ligands of the virus and the receptors of the host cell. Once the host cell becomes infected, the virus will gain entry into the cell. This could happen by endocytosis, or it could happen if it's an envelope virus by fusion of the envelope to the cell membrane. And then they will fuse, and then the membrane of the cell will actually go around the virus, and then the virus can move into the cytoplasm. And then if it's a DNA animal virus, that DNA will actually move into the nucleus. It will be transcribed as viral messenger RNA, which will move into the cytoplasm and be translated into proteins, viral proteins, including capsid proteins. In the meantime, the viral DNA will be replicated in the nucleus. And then the capsid proteins will assemble with the newly replicated DNA to form the new virion. And then this virion will be released from the cell either by lysis of the cell, which the virus will cause the lysis of the cell, releasing the new virions, or by the budding life cycle. And I do need to mention that I'm mainly talking about these two life cycles, but there are a few other life cycles of viruses. And we'll talk about the latent life cycle of viruses in a little bit. In the latent life cycle, the virus actually doesn't replicate, and we'll talk more about that. But there is another viral life cycle I will briefly mention. It's the not peripheral, what is that called? Drawing a blank on the name of that. A word starting with P that means it's a long-term virus, which is shed over a long period of time. Persistent, there it is, persistent viral infection. There's a persistent viral infection life cycle, which is another one. And we're only going to mention that in a sentence. So the two main I'm going to talk about are the lytic life cycle and the budding life cycle, which are most animal viruses. Any question about any of that? Anyways, the virus will be released from the cell, and then it can once again attach to the cell and follow the same life cycle. Any question about the multiplication of DNA animal viruses? All right, let me blow this slide up. It's showing you the RNA family of viruses, and there's more of them. I believe there's 12. 1, 2, 3, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Didn't remember right. 15. 15 RNA family of viruses that cause human disease. You don't really need to memorize this slide. I'm just showing you the different animal viruses that cause disease. And there's the animal family of viruses, and they're all RNA viruses. And the diseases that they cause, shown in this slide here. You don't need to know the strand of DNA, whether it's double RNA strand, single negative strand, single positive strand, whether the virus is enveloped or naked. Picorna viridae, I do remember that one. It's a naked virus. And where is coronavirus? That one I know too. It's an envelope virus. And you don't need to know the capsid symmetry or the size of the capsid. Any question about any of that? All right. RNA, let me back up a minute. RNA family of viruses as a rule of thumb, replicate in the cytoplasm. Why? Because they're an RNA virus, and the machinery for replicating, well, at least translating RNA, is in the cytoplasm. So the RNA family of viruses, as a rule of thumb, replicate in the cytoplasm. We're only talking about animal viruses, by the way. Although it is true that bacteriophage viruses, which have RNA, do replicate in the cytoplasm, but that's true of all bacteriophage viruses. There is one exception to this rule of thumb, and you should know it. Retroviridae, the retroviruses, are an RNA family of viruses, meaning they have an RNA genome, but they do not replicate in the cytoplasm. The retroviridae replicate in the nucleus, and that's the only exception to this rule of thumb that I know. Any question about any of that? All right, here's some different RNA containing animal viruses. You can note that they are different sizes, different shapes. And of course, there's more of them, families, than the DNA family of viruses. The replication of RNA viruses is similar to the replication of DNA viruses, with some differences because we have RNA as our genome instead of DNA. The viral replication occurs, as the rule of thumb, like I stated, in the cytoplasm, with the exception of the family Retroviridae. Some examples: positive strand, single strand RNA animal virus, and a negative strand RNA animal virus replicate by using this strand to make the template strand. So the positive strand makes a template, which is a negative strand, and then the negative strand is used to make more of the positive strand. The same thing with the negative strand. It will make the positive strand and then use that positive strand to make more of the genomic negative strand RNA. Any question about any of that? Now, for a double-stranded RNA virus, the two strands of RNA just need to separate, and then you use one strand as a template to make the other strand. And that's how double-stranded RNA viruses replicate. Any question about RNA multiplication? RNA viral multiplication, I should say. So for DNA, we can transcribe that into making messenger RNA, and then translate the messenger RNA, viral messenger RNA, into the viral protein. For a positive strand, single strand RNA virus, it can serve as messenger RNA, meaning the positive strand, single strand RNA genome can be read by the ribosome and be translated into viral protein. The negative strand RNA virus cannot be read by the ribosome, and it is not translated into viral protein. How the negative strand RNA virus makes viral protein is it makes a copy of this negative strand, making the positive strand, and then that positive strand RNA can be used as viral messenger RNA to translate into viral proteins. And then, of course, that positive strand RNA can then be used as a template to make more negative strand RNA, and that's the genomic RNA. Of course, the genomic positive single strand RNA makes the negative strand RNA and then uses the negative strand RNA to make more genomic positive strand RNA. And that's how viral multiplication and transcription and translation occurs. Any question about any of that? All right, let's talk about animal viral RNA replication. For an animal RNA virus, if it's the negative strand RNA virus, let me blow that slide up a little. Whoop. It will replicate by making the positive single strand RNA and then use that positive single strand RNA to make more of the genomic negative single strand RNA. Then the positive single strand RNA will be translated by the ribosomes to make the capsid proteins and other viral proteins, which will then get together with the newly replicated single strand negative strand RNA to make the progeny virions. For the animal virus, which is a positive strand, single strand RNA virus, it will make negative single strand RNA from the positive strand, and then use the negative strand as a template to make more of the genomic single strand positive RNA. And then that single strand positive RNA can be read by the ribosomes to be translated into the viral proteins, which will then get together with the replicated viral genome to make the progeny virions. For the example of a positive single strand RNA virus, how it multiplies in depth is the RNA virus will bind to the host cell membrane. Once again, there's specific ligands on the virus that binds to specific molecules on the cell, which will be the cell membrane that we call receptors. And that determines the range of cells that this virus can infect. The virus will be brought into the cell either by endocytosis, or if the virus has an envelope, it can fuse with the cell membrane and then be brought into the cell. The virus positive strand RNA will make the negative strand and then use the negative strand to make more genomic positive strand RNA, genomic RNA for the virus. And this is happening in the cytoplasm. I do need to mention that. The positive strand RNA virus can be read by the ribosome. That can be either the original positive single strand RNA or the replicated, newly replicated positive single strand RNA. Can be read by the ribosome to make the proteins, including the capsid proteins of the virus, which will then get together with the newly replicated genomic RNA positive single strand to make the new virions, and they will leave the cell either by lysis of the cell or by budding of the virus out of the membrane of the cell. Any questions about the multiplication of a positive single strand RNA virus? All right, the negative antisense single strand RNA virus will once again bind to the receptors of the cell membrane and then enter the cell either by endocytosis, or if this is an envelope virus, it can enter by fusion of the viral envelope to the cell membrane. The virus will get inside the cell, and then it has to be copied to make the positive strand, single strand RNA virus, which will be used as a template to make the genomic strand, the single strand negative strand RNA virus. The positive strand RNA virus will then be used by the ribosomes to transcribe the viral proteins, including the capsid proteins. The capsids will get together with the newly genomic replicated negative single-strand RNA virus to make the new virions. And let me state that both the translation of the positive strand and then replication of the negative strand RNA is happening in the cytoplasm. And then the newly replicated virus will be released from the cell, either by lysis of the cell in the lytic life cycle or by budding of the cell in the budding life cycle. Any question about the multiplication of negative strand antisense single strand RNA virus? All right, let's talk about the exception, and that's the family of Retroviridae, the retroviruses. They are RNA family of virus, but they replicate in the nucleus, not in the cytoplasm. The retroviruses include the lentivirus, which include HIV-1, HIV-2, which cause human AIDS disease, and then also it includes feline leukemia virus, which causes leukemia in cats. And the cats sort of waste away from the leukemia, similar to AIDS and their immune system, because of the leukemia deteriorates, and then the cats tend to get in, what do you call that? Get numerous diseases because their immune system is very weak. So feline leukemia virus is sometimes called kitty AIDS, but remember, it's not really AIDS, it's a leukemia. Retroviruses contain a unique gene, and this gene is the enzyme for reverse transcriptase. Reverse transcriptase is an RNA-dependent DNA polymerase. And I doubt that means a lot to you, so let me tell you what it means. I don't know why that's so small. Try and blow that up a little. There we go. So we have single strand RNA. And then you don't really need to know this, but it's actually a positive single strand RNA. Let's see if I can get that. There we go. Oh, wrong one. That's not the right one, is it? I'm not seeing how to do text on this. Oh, there it is. And let me see if I can get an arrow in there. There we go. I should probably copy that. Oop, don't want that. Back. There we go. We'll use that in a minute. This is going to happen with reverse transcriptase. I'm just going to abbreviate that RT. Let's see if I can put that here too. We'll talk about this in a minute. And what the reverse transcriptase makes is single strand DNA from RNA. And then reverse transcriptase. Let me see if I can copy that. Never mind. Just go here. Reverse transcriptase can then work on single strand DNA to make double strand DNA. And that's what reverse transcriptase does. Any question about that? I think your book does talk about reverse transcriptase making DNA from single strand RNA, but in reality, it happens in two steps. First, it makes single strand DNA, and then it makes double strand DNA from single strand of DNA. Any questions about any of that? And you can see why retroviruses replicate in the nucleus, and that is they have a DNA intermediate in the replication of the virus. Now, what happens next? Let me go ahead and see if I can put that in there. Nope. Yeah, it's still there. We then make this. Let's see if I can just select that. Double strand DNA can be transcribed to make positive single strand RNA, which is the genomic RNA virus. And of course, the positive strand can be read by the, let me just do an arrow. Can be read by the ribosomes to make proteins. The proteins will then, the capsid proteins will then get together with the in fact, it's all of this actually. So let's go ahead and make that. Meaning the genomic RNA can get together with the proteins to make the new virions. Which will then bud out of the cell. All the retroviruses follow the budding life cycle. And then that's how the cycle come on. Continues. Any question about any of that? So that's how retroviruses replicate. Reverse transcriptase actually gives the retrovirus, family retrovirus, its name. The RE from reverse is in retrovirus, and then the TR of retrovirus comes from transcriptase. Any question about that? Here we're looking at a retrovirus, and you can see it has spikes. It has an envelope. There's the capsid, and there is the viral genome. You don't need to know this, but in HIV, it is 2 copies of the positive single-strand RNA. You only need 1, but at least HIV, there are actually 2 copies of the single-strand positive strand. And then there's 1 copy of reverse transcriptase. Let me see if I can blow that up and see it. It's this protein right here. So all of that is inside the virus. So with multiplication of retroviruses, attachment, penetration, and encoding of the retroviruses is basically like the same for animal viruses, but the biosynthesis stage is different. And that is the RNA positive single strand is made by reverse transcriptase in 2 steps. Your textbook only shows it in 1, but in reality, it's made in 2 steps into double strand DNA. So first it'll make single strand DNA, and then the reverse transcript will take the single strand DNA to make double strand DNA. The double strand DNA can then integrate into the host cell chromosome, and it can be transcribed into positive single-strand RNA, which is the genomic RNA of the virus. It is also the RNA that can be translated by the ribosomes into the capsid proteins. The capsid proteins will get together with the newly replicated positive strand genomic RNA to make the progeny virions, which will then bud out of the life cycle, bud out of the cell. So a retrovirus will once again bind to the cell membrane with specific molecules that we call either ligands or adhesions, depending, I guess, on who the author is of the textbook. And it will bind to specific receptors, which are molecules on the cell membrane that the ligand or adhesion binds to, and then be brought into the cell. In the case of retrovirus, the virus will be brought into the cell by fusion of the envelope to the cell membrane. Once in the cell, the viral genome, which is positive single-strand RNA, will be made in 2 steps by reverse transcriptase into first single-strand DNA and then double-strand DNA. The double-strand DNA will then move into the nucleus, and it will form a provirus, meaning it will bind to and insert itself into a host chromosome. Once that happens to this cell will always have the retrovirus inside it as a provirus. So once a provirus is made for an animal virus, that provirus will remain for as long as this cell is alive. And when this cell would divide into 2 cells, the provirus will be replicated with the rest of that chromosome. The provirus can be transcribed to make RNA, and this will be the positive single-strand RNA, which is the genomic RNA of the virus. That positive single-strand RNA can be read by the ribosomes. It'll move into the cytoplasm and then be translated in the cytoplasm into viral proteins. And the viral proteins will get together with the viral single-strand RNA, positive single-strand RNA, to make the new virion, which for a retrovirus will bud out of the cell. For HIV, it'll bud out of the cell membrane. And then that continues the life cycle of the virus. Any question about any of that? Remember, usually when a budding virus buds, it does not, most of the time, it does not kill the host cell. If this host cell dies, it's because the immune system recognizes that the cell is infected, and the host immune system will kill this cell. The virus usually does not kill the cell. Any question about that? All right, let's talk about a little bit about Coronaviridae. This is a family of virus that actually I never talked about until COVID-19 came about. This is the family that contains COVID-19. And if you say COVID-19 is coronavirus, that's not quite correct because there are other members of the family. COVID-19 is only one species in the family Coronaviridae. This family is single strand, positive strand RNA virus. They're enveloped. And this family actually causes the second most common cause of the human cold. Remember, the third most common cause, or the least most common, is adenoviruses, and that's a DNA virus. This is an RNA family virus, and it's the second most common cause of the human cold. So this family was well known to humans long before COVID-19 came around. And you'll note that this is a picture of coronaviruses, and I've used this as a template for my microbiology class for many years before COVID-19 ever showed up. So this is a coronavirus. It's actually not COVID-19, although COVID-19 looks like this. Any question about any of that? I just thought it was a cool looking virus, so I chose it. And then, like I said, I became useful because I do talk a little bit about COVID-19 now. Besides COVID-19 and the common cold, coronaviruses can also cause SARS, MERS. SARS, MERS, and COVID-19 are all similar, and that is they tend to cause severe acute respiratory syndrome. And that's actually what SARS stands for, severe acute respiratory syndrome. MERS stands for Middle East Respiratory Syndrome, and the patients do get severe acute respiratory syndrome. With COVID-19, some patients get severe acute respiratory syndrome as well. Middle East Respiratory Syndrome, or MERS, has the highest mortality rate. People who become infected with MERS, about 36% of them will die. Fortunately, MERS does not spread from person to person. People who get MERS, almost all of them, are people who are in close contact with camels. Apparently, MERS is a virus of camels, and occasionally it comes out of the camel, meaning replicating the camel, and then can infect a human. But you have to have really close contact with camels, and that would be something like a camel breeder or a camel rider. SARS also is more lethal than COVID-19, and about 10% of patients die from SARS. SARS is a virus which first made its appearance in the human population in 2003. And fortunately, they got control of that virus and it did not spread further. It reappeared one time later. I don't remember the year, and it really only infected China when it came out the second time. And fortunately, it never spread further. And at the moment, SARS is extinct in the human population. And it's a good thing because it's more lethal than COVID 19. One reason why SARS, they were able to control it is with SARS, everybody who gets infected has signs and symptoms of disease. With COVID 19, that's not the case. And if you remember, President Trump actually put what do you call that? A quarantine on countries like China who had COVID 19 in it and did not allow them to enter the United States in an attempt to keep COVID 19 out of the country. However, COVID 19 did come into the country and probably by an asymptomatic person. COVID 19 can be asymptomatic, and in young people, young adults, children, and teenagers, about 50% of people who get COVID 19 will be asymptomatic. And that doesn't mean that they can't spread the COVID 19. And of course, the danger is they could spread it to a child's grandparent or a young adult's parents, in which case COVID 19 could cause serious disease. COVID 19's mortality rate initially was between 0.2% to 3%, but after that variant, what was the name of that? Losing the name of that variant of COVID 19, which was very common, which is actually 2 years ago, maybe even 3 years ago now. After that variant came out, the mortality rate of COVID 19 has dropped by about 50%. COVID 19 spreads more easily than the flu, and one person is likely to infect between 1 and 2.5 other people, which is why COVID 19 has continued to spread in the human population. Omicron, that's the variant. After the variant Omicron came about, COVID 19's mortality rate has dropped by about half. So it's something between 0.1% and 1.5%. And I think that's in the United States, by the way. The reason why the later strains of COVID 19 have about the same mortality rate as Omicron is because all of the later strains of COVID 19 are derivatives of Omicron. The most lethal strain of COVID 19 was the Delta variant, and that was actually the strain of COVID 19 before Omicron came about. Any question about any of that? All right, COVID 19 is a shorthand name for the virus. It does have a scientific name, and it's called Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2. However, this name is so close to SARS that even among scientists, they tend to call it COVID 19 to distinguish it from SARS. COVID 19 actually stands for covirus. That's what the COV stands for. And then ID stands for it was identified in 2019. That's where the 19 comes from. So that's what COVID 19 means. Coronavirus ID'd in 2019. In 2019 was when, late in 2019, when the virus first became apparent in China. COVID 19 is closely related to bat coronaviruses, and it likely originated from bat coronaviruses. They've sequenced COVID-19 and some other coronaviruses, and they found that it was actually a recombination, COVID-19, of two bat coronaviruses and one pangolin coronavirus. The pangolin virus only is about 10% of the genome of COVID-19, but it's very important because the pangolin genome codes for the spikes on the COVID-19, and this is why COVID-19 can infect human cells and why it's so contagious, because of the pangolin genome of COVID-19. The COVID-19 probably was in bats and then went to an intermediate, probably a pangolin, before it was introduced into humans. So probably the first human who got COVID-19 got it from a pangolin. And I think it was the city of Wu in China. Somebody can correct me if I'm wrong or mispronouncing it. The reason why it showed up in that city is because in that city and other Chinese cities, they had a whole bunch of animal markets, which vendors would sell different animals as well as other products to sell in the market. And then there's a whole bunch of people in the market. And likely either the person who was selling that pangolin or the person who bought that pangolin was likely to be the first person who came down with COVID-19. Any question about any of that? By the way, China has noticed that now 3 times out of the animal markets in China, they've had 3 lethal viruses come out and enter the human population since 2003. The first was SARS in 2003. The second one was also SARS. I don't remember when it was, sometime after 2003, but before 2019. And then COVID-19. And the Chinese have realized that having all of these exotic animals like pangolins in the market, and then lots of people in the market, that it's more likely for an animal virus to spread from the animals in the market to a person. And so the Chinese have started, or for several years now, have shut down not the animal market, but shut down the selling of exotic animals in the animal market to try and reduce the chance of a lethal virus entering the human population. Any question about any of that? All right. COVID-19 is transmitted primarily via droplets from coughs and sneezes, but it can spread from other things like people breathing, especially singing tends to spread COVID-19, or talking, not highly talking, like you're unlikely to get it just by talking to someone, but you are likely to get it if somebody's singing near you when they have COVID-19, or somebody sneezes or coughs. You have to be within a range of about 6 feet. It can spread further than 6 feet, but you're less likely to get infected if you're further than 6 feet away. It can also spread by indirect contact via contaminated surface, like somebody sneezes on their hands and they touch a doorknob or a light switch. Somebody else coming later can pick up the COVID-19, get it on their hands, and then inoculate the virus into a mucous membrane in their face. Fortunately, with COVID-19, for whatever reasons, it does not tend to spread that way. It tends to spread via droplets from the air, like a cough or a sneeze. but it can survive up to 3 days on plastic or metal surface, or a day on cardboard or paper surfaces. That's not in the sunlight, by the way. Sunlight has UV light in it, and that's actually a good way to kill COVID-19. Put the potentially viral infected thing in sunlight, and after about 15 minutes, depending if it's summer or wintertime, it takes longer in the wintertime, less time in the summertime, but after about 15 minutes, COVID-19 will die from UV light. And you could always leave it for a day if it's on cardboard or paper or metal or plastic for 3 days, and COVID-19 will die. It turns out it has a lipid membrane and the envelope, I should say, and the envelope can dry out after a day on cardboard or paper or 3 days on plastic or a metal surface. And the virus will not be contagious if the envelope dries out. Symptoms of COVID-19, they're mostly flu-like. Initially, runny nose and sneezing were less common. Actually, nowadays with the new COVID-19s, runny nose and sneezing are very common. Shortness of breath and deaths are more common than the flu. Mortality rate increases with age of the patient. The ill health of the patient increases with men over women, and also increases with Blacks and Hispanics. It's not known why Blacks and Hispanics are more likely to get a severe disease or die from the disease than whites and Asians, but it is verified in the United States epidemics, or not epidemics, what am I talking about? Epidemiology, and Canada, Canadian, and Mexican epidemiology. The incubation period, you need to be exposed to COVID-19 for 2 to 14 days before you will get the first signs or symptoms. And you should realize that young people, about 50% of them will be asymptomatic. They will never develop any signs or symptoms of having COVID-19, but they can still get it and spread COVID-19. Prevention, use correct hand washing, keep your distance from other people, especially if they're coughing or sneezing. Don't touch your face with your hands until you've washed your hands or sanitize your hands. And then, of course, wear a mask. That will prevent you from spreading COVID-19, and it reduces your risk of picking up COVID-19. When you're sick, it's best to stay home, cough into your tissue or elbow, and then don't spread COVID-19. You stay home, only people in your home can get it. Any question about COVID-19? All right, let's move on to viruses and cancer. Some human cancers, actually 10 to 15% of human cancers, are caused by viruses. It's well known that human papillomavirus can cause cervical cancer and skin cancer, and then hepatitis B virus can cause, and hepatitis C virus can cause liver cancer. However, not all cancers are caused by viruses. For example, Tang in 2013 found that no breast cancer or glioblastomas were caused by viruses. Any question about any of that? Cancer is uncontrolled growth of cells. That's all I'm going to talk about that. Viruses can cause cancer in two main ways. The first way a virus can cause cancer is the virus can integrate into the host cell's chromosome, forming a provirus. That will cause a permanent genetic change in a cell. For example, if the provirus integrates into a tumor suppressor gene, that will turn off the tumor suppressor gene. we're not going to talk about what the heck a tumor suppressor gene does, but it actually suppresses the formation of a tumor. Okay, that's why it's called the tumor suppressor gene. And then the rovirus can also mutate other genes of the cell, and the mutation can lead to the formation of a cancer. So the DNA viruses and the retroviruses can cause cancer because they integrate. If they form a provirus, they can integrate into the host cell chromosome and then muck up the cell's DNA, leading to the development of a cancer. Any question about that? The other way a virus can cause cancer is the virus can contain an oncogene, and these we call oncogenic viruses because the virus itself has a gene on it that we call an oncogene, and this gene tends to cause a tumor to form. An oncogene is actually a mutated form of a normal gene that is involved in cell growth. So the normal gene normally helps a cell grow, like go through cell division to make more cells. This is a mutated gene, and then it causes the cell to replicate, making the cell much more likely to become a cancer because the gene causes the cell to keep growing unregulated, which is by definition cancer. These mutated oncogenes help transform a normal cell into a cancerous cell. And so when a cell becomes infected with an oncogenic virus, that oncogenic virus can contain an oncogene, which can then change the cell, causing it to grow uncontrollably, which will increase the chance that a cancer will occur. Any question about any of that? All right, those are the two ways that viruses can cause cancer. Muck up the DNA by forming a provirus, and then bring an oncogene into the cell. Any question about any of that? All right. I mentioned that there were two main ways that an animal virus can replicate. That is the lytic life cycle and the budding life cycle. There are two other ways that an animal virus, well, I should say two other life cycles that the animal virus can have. And that's the latent life cycle and the persistent viral infection. I had to change my wording there because a latent life cycle, the virus doesn't actually replicate while the virus is in the latent life cycle. In the latent viral life cycle, this is usually a budding virus. The virus goes latent. The virus remains asymptomatic and then stays around in the host cell for long periods of time. And while it is latent, it does not cause disease, does not cause the patient any signs or symptoms. In fact, the virus is dormant when it goes latent. Sorry, not virus. When the virus goes latent, the virus is dormant, not active. The trouble is the latent virus can then later become active, such as when you have cold sores, herpes simplex virus type 1. While you have a cold sore, the virus is in the budding life cycle, but then the cold sore goes away, and that's because the virus has entered the latent life cycle. And then you can get a cold sore again later. And what happens is the virus leaves the latent life cycle and then enters the budding life cycle again. And the virus is actually replicating in the budding life cycle, not in the latent life cycle. And while it's budding is when it causes disease such as the cold sore. There's usually a trigger that we call the stressor for the virus to leave latency. And for cold sores, the stressor is usually the patient comes down with a cold. And that's why we call it a cold sore. Any question about any of that? Another case of a virus that goes latent is the virus that causes chickenpox. When the child or adult has signs or symptoms of chickenpox, the virus is in the budding life cycle. The virus is actively replicating, causing disease, causing the chickenpox. And then the chickenpox goes away, and what happens is the virus enters the latent life cycle, hiding out, and it can remain latent for decades. And then the virus can become active, and it doesn't form chickenpox the second or any time after the first. It only forms shingles. So every other time the virus becomes active, the patient will get shingles. The first time, the patient will get chickenpox. And in between the shingles and the chickenpox, the virus is latent, not replicating, not causing disease signs and symptoms. The trigger for the shingles is usually the patient becomes old in age and their immune system decreases. And that's usually when shingles comes out. Any question about any of that? Oh, the persistent viral infection is a naked virus, but it does not lyse the cell. So it does not follow the lytic life cycle. What happens is the virus replicates in the cell, and then it leaves the cell in a vesicle. So the virus is incorporated into a vesicle. The vesicle then moves to the cell membrane, fuses with the cell membrane, and then dumps the virus outside the cell. So it's a naked virus. It typically does not kill the cell when the virus replicates. The virus replicates usually at low numbers, similar to the budding life cycle. And then when it replicates, like I said, it doesn't kill the cell. So it sort of follows the life cycle similar to the budding virus, except it leaves the cell in a vesicle, not in a bud from the cell membrane or the nuclear or endoplasmic reticulum. It leaves the cell by the vesicle. And usually the vesicle will fuse to the cell membrane and then dump the contents outside the cell. Any question about a latent virus and a persistent viral infection? Now, remember, a latent virus, the virus is not replicating in the persistent viral infection virus life cycle. The virus is replicating, but at low numbers. Persistent viral infection is a disease which occurs over a long period of time. Usually it starts low, mild signs and symptoms, and then progresses over time, where the viral load builds up over time, getting bigger and bigger. The virus might never be cleared by the host immune system, and sometimes a persistent viral infection can be fatal. Like in the old days, when the patient had HIV and the virus was replicating and then caused AIDS. In the old days, before modern medication, the AIDS patients tended to die. And so HIV, like in the first 1980s and 1990s, tended to be fatal, a persistent viral infection that once the patient had the infection, they always had the virus. Another example is subacute sclerosing panencephalitis, which actually is caused by the measles virus, and it can be fatal, causing a persistent viral infection, which starts mild, gets worse and worse over time. The viral load builds up and then can become fatal. However, some persistent viral infections are tolerated, like cytomegalovirus. Usually, with that one, the disease will be mild and then will get worse, and the viral load will increase, but it will plateau and not get worse than a certain level. And that's usually what happens with cytomegalovirus. Varicella zoster, causing chickenpox and shingles. My mother had a persistent viral infection caused by varicella zoster. When she got shingles, she was one of the unfortunate individuals where the shingles never went away. She had shingles from when she got it to the rest of her life, which was something like 3 or 5 years. I'll have to think about it. But she never had it, never went away. Like about 1 in 1,000 people will get shingles like that. So it's very rare. Some other herpes viruses can be tolerated, where the patient gets it and they have it for a long time. Any question about any of that? All right. Let's talk about the different types of a viral infection. Actually, we're out of time, so I think I'll stop here and ask, are there any questions?
I have a quick question.
Mhm.
Do we have a quiz today? It keeps showing up on the calendar.
Is it quiz 5C?
Yeah.
Yeah, that's not a quiz. That's an assignment. It has to be an assignment so that I can give you points. I'm going to change quiz 5, so it will be eventually worth 75 points. But I can't give it to you now because then you'll have too many points from the quiz. You get to throw out one of your quizzes. So I made quiz 5, it's close to 0 points, but I will change it so it will be worth 75 points. And then I'll subtract that, your lowest quiz, in the drop quiz column.
Okay, so we don't have to do anything with it?
No, there's nothing you can do with it.
Okay, just wanted to make sure. Thank you.
All right. Any other questions? Like I said, I'll be in the lab for 15 minutes. If you don't have questions, you don't need to show up in the lab. All right, if there's no questions, I'm going to end it here and I'll see you next time.